Hello, students. Today our topic is antibody structure. We know these antibodies are the gamma globulins, or antibodies are the proteins, and they are produced by the B lymphocytes. So in it, we see that the structure of antibody is that it have these two heavy chains and two light chains. So this is the heavy chain, second heavy chain. So you can see these two heavy chains. And here in it, you can see that I am writing it in the four parts: first, second, third, fourth. So this shows that the first part is a polypeptide part made up of 110 amino acids. The second part of the protein is made up of 110 amino acids, and the third is made up of 110 amino acids, and the fourth part is made up of about 110 more or less. So, and these two heavy chains are the same gene product, and they are connected with help of disulfide bridges with each other. So these disulfide bridges are connecting these two heavy chains with each other. So, and the other protein part of this antibody is the light chain. And this light chain is making this and this part. So you can see that there are two parts of the protein, and basically four polypeptide chains are coming together, two light chains and two heavy chains. And this part is this antigen binding fragment, and this part, the antigen binding fragment, that is FAB. So this one is the FAB part, and this one is FC part. So this part, that is FAB part, is the variable region. Variable region. And this part is involved in the binding with the epitope or antigenic terminant. And this part is termed as FC part, crystallizable fragment, or you can say constant fragment. So there are two parts of the antibody, and these light chains are bonded with the heavy chains with the help of disulfide bridges, and these heavy chains are also bonded with the help of disulfide bridges with each other, and then we get this Y-shaped antibody structure. So it have these segments of 110 amino acids, 110 amino acids, just like in the heavy chain. The light chain also will have this 110 amino acids and 110 amino acids. It means This light chain will have two parts, that is variable region, which consists of 110 amino acids, and the constant region, which will be made up of 110 amino acids. The heavy chain will have 110 amino acids, the variable region, and 330 more or less amino acids, which will make constant region of the heavy chain. These two light chains and two heavy chains, they will come together and they will make this antibody structure. So, but the antibody structure, if we see that if I take this light chain as, it should not be simple because our system can produce about 100 million different types of antibodies. So it's not so simple. So each variable region will have different this amino acid composition, therefore it will have different structure. And this light chain, so it's the globular protein, and this heavy chain will have different amino acid sequence, therefore it will give a different structure. So in both sides, so it is not so simple. It should be something like this. Complex proteins with different structures, with different amino acid sequence, especially in the variable region. So it's not should be simple Y-shaped structure. But anyway, we are thankful to the Edelman and Porter, the two Nobel laureates who have derived and reduced the antibody structure, which is in accordance with the new findings and this Y-shaped structure. And they got Nobel Prize for their contribution. Anyway, antibodies are these gamma globulins which are produced by the B cells and secreted by the B cells, especially plasma cells. And it consists of two heavy chains and two light chains which are bonded with each other with help of disulfide bridges, and it is have two parts. That is, it one part that is this part, and other one the binding region. So it will have two binding arms and one Fc part. So basically the Fc part is the part which is performing the functions of the antibodies, that is marking the or opsonization, marking the pathogen, and to inform the immune system and also to activate the complement system. So this part is the FAB part is just to bind specifically with the target pathogen. Okay, thank you. See you then. Allah Hafiz.